Good afternoon, everyone.
Good afternoon, sir.
Yes, so we'll start the class now. In the beginning of this course, we started about how the future of humans, or how problems that the human species had, develop a motivation to or find a motivation for why studying stem cell biology is important. We noticed, we discussed about the why these bad mutations are accumulating and how the social fitness is preferred over biological fitness, and thereby how the population, the biology or the genome is getting accumulated with bad genes or mutated genes. And moreover, we also have unnecessary exposure to chemicals, physical, the problem of unnatural selection and improper mate selection. So there will be problems, and we have to decide on how we should look like. On one side, as bioengineers, we try to develop prosthetics and other robotic things or non-inanimate mechanical devices, electromechanical devices, to aid in our deficiencies. But at the end, I think we will prefer a an organ replaced with another organ, right? So while both are two approaches, and stem cell biology is the approach towards trying to regenerate or provide biological solutions, biology-based solutions to the problems of future problems to human species. Later, we discussed about the early embryology stages and how development from a zygote to different organs and cell types happens. We discussed about zygote and then blastula with its inner cell mass. And then we discussed about gastrula, where the three primordial germ layers form: ectoderm, mesoderm, and endoderm. And there is also one more thing called as primordial germ cells. So the primordial germ layers will make what we are.
So,
Voice.
Yes, yes.
your voice is very low that it's not audible enough.
Okay. Let me try again then. I thought I fixed the problem before starting the class. So, is it any good, or what is the problem? Is it still not great? Just tell me so I'll try to see if I can. One second.
It's only a bit normal, so it's not like as clear as it used to be.
Yeah. Okay. I'm at college now. That's one of the problems. Mhm. I will try this and let me know. Is it any better now, or is it still same? Can you hear me?
Yes, sir.
Yes, sir.
It's kind of clear now.
Is it a bit clear now? Can you confirm that, please?
But there's an echo.
Echo. Okay. How is this now? Can you say, is the audio same or is there echo? Anybody?
There's no echoes and all. It's better.
What we were using before. Let's go ahead with this, and in case if there are any issues with the video, I will share with you at a later time. Okay.
Yes, it's better now.
So I don't know where, I'm unsure where you lost, but I'm just reviewing what I have already discussed before. Okay. So primordial germ layers will give rise to all the somatoplasm, and somatoplasm is every part of our body apart from the gametes or gamete forming cells, which is the germ plasm. Germ plasm arises from primordial germ cells, and primordial germ layers give rise to the somatoplasm. So in today's class, and we in today's class we have also discussed some of the things I wanted you to remember about zona pellucida, cortical reaction, about details about spermatogenesis and oogenesis, and the sequence of growth, zygotes, cleavage, upon cleavage, morula, blastula, glastula, neurula, and fetus, and so on. So what we are going to start now is the next lesson, or actually this is the first lesson as per the syllabus. It is, I told you that in unit 1, it is the most important one, and it is most easiest one. It is the easiest one if you are planning to score well. Okay, and this the first chapter is like the summary of the whole syllabus. Okay, so I would I would encourage you all to download this. This paper is given already in the syllabus. You please go through it and download the first paper. And then start reading it. So it is not a typical textbook. It is a review. So there will be others understanding of the current developments in this field, and they also put in their perspectives and what to expect in the future and so on. The objectives of this paper are, for me and you, is to know about the definitions of stem cells and trying to find out and trying to understand their properties. And then we will also try to learn about embryonic and adult stem cells, and maybe a little bit about cord blood stem cells as well. And eventually we will go on to see some of the methods about how to derive human embryonic stem cells. Okay. So I will just take a step back and try to understand what is life. To define life is a difficult thing. In the last class too, I think we may have seen this picture before. So there are some essential properties where you see, this is called, they will say as Mrs. Gren, to remember it easier. Movement, respiration, sensitivity, growth, reproduction, excretion, and nutrition. Not that every organism shows all of these, but there will be a majority of them. There is one that is shown, that property is shown by all living organisms, and that is reproduction. Right? Those that reproduce more will be prevalent, and they are likely to exist longer in this on surface of Earth. If you see growth, you can see plants, animals, protozoa, and bacteria also grow. But viruses, there is nothing called growth. They are assembled within the bacterial cell, like we saw in the lytic life cycle in lambda phage, for example, in the previous semester. As I already said, reproduction is present in all. Reproduction and replication. Replication forms the basis for reproduction eventually. And you can think of it, there is no replication, there is no reproduction. That's how it is. And what we are concerned of one of all these properties as per this syllabus, this course, is repair. One important thing, if you see a plant loses a small branch, it can regrow a different branch. Or if there is a hole in the stem, there is somehow, it can, to a degree, it can repair itself. How many of the times we played in the fields or outside or at school, we fell down, we had wounds on our knees, and our skin repaired the wounds, right? So you can think of all the uh, think of so many injuries that happen to every organism, and you can see a certain degree of repair in almost all of them. But there are some things very good. We lose a hand, we cannot regrow a hand, isn't it? But there are some animals which can regenerate, dramatic dramatic regeneration capacities they have. You may have noticed the lizard with the lost tail, and then eventually it will get to regrow a new tail, right? So this one is, it is cut off here, and this part is grown new. It's a gecko. In the house lizards that we see, we sometimes see the whole body and a stumpy tail. If a predator is holding it here, it just kinds of loses its tail and move forward, and then it can regrow a tail later at a later time. Similarly, there are some animals like gecko which can regrow a whole limb. That's important. That's something fantastic. Within about 3 to 9 weeks, it can grow a new limb itself. If you take planarium, it's flatworms. You have the head, the body, and the tail. And if you were to cut each of these into 3 pieces, the one with the head will regrow the body and tail, and the tail will regrow a whole head too. This is incredible that an organism can cut itself, or I mean, when it is cut or broken into pieces, it can regrow into many, each one as an individual organism. Then they all have the same genome, and they can be referred to as a clone, isn't it? It's a form of kind of asexual reproduction. It's not necessarily happening deliberately, but if there is a breakage in the organism, it somehow regenerates the rest of the body. And that's fantastic. That means while as humans, we don't have those capacities. But what you are seeing is biologically cells may be able to do that. With increasing complexity, say fish, reptiles, flatworms come even before invertebrates, and then we have birds, mammals, and apes, and so on. The regenerative capacity actually reduces. Sea anemones are there. If you split them into two, they can grow into two different sea anemones. Same case with the starfish as well. Okay. So what happens in higher animals too? There is certain degree of regenerative capacity, like I discussed about the skin, for example. And our wounds, all the healed ones, healing of the wounds. There is something in Greek mythology. In our Indian mythology too, we have a lot of these kinds of stories. There is a person called Prometheus. Because he committed something, he was chained to a mountain. The mountain is called as Caucasus, I think. And this guy is the Prometheus. And there is an eagle that comes to the person every day and eats the liver of that person. And overnight, the liver regrows for the next day. Every day, it keeps on doing it. What's the coincidence between this mythological story and human body is that apparently liver is the only organ that actually can, with maximum capacity for regeneration within the human body. What I mean is, you can take away as much as 75% of liver, cut it away, and you just leave 25% of it. It can, over a period of time, regrow to 100% liver, right? You cannot talk about this any other organ like this. Say if somebody is donating liver to somebody else, the donor is likely to regrow the whole liver. You cannot say the same thing about heart transplant, or heart transplant, you cannot do that because we only have one. Heart donation, I meant. But kidney donors, if they give away one kidney, they have to live with one kidney alone, right? So this is a nice trying to find coincidences with mythology and real biology. Or did the ancient Greeks already know that liver is the only organ that is actually growing? It's unlikely, but that's a coincidence. I have a question here, and the question description is, the plant is something like this, and it is a tall, high about 2 meters. And you or I have put a nail about a distance of 1 meter high from the ground. After about 10 years or so, the tree is now grown into huge, very tall, and it is about, total height is about 8 meters. Where would you find this nail in this tree? That is a question.
Same 2 meters above ground.
Yeah, why is that?
Sorry, it's 1 meter above ground because
Yes, yes, I understood
uh,
that.
the
Why?
top, only the top of the tree grows. Only the extremities grow in a plant.
That's right. As you may have learned from your 10th class or plus one, plus two, and all, the growth in different organisms is different. Compare the growth of a plant. The plant grows by the tips. You can have the tips of the branches, and they have what is called as apical meristem. And there is also another layer. If you make a cross section of it, you have uh, can anybody recollect? You have these bundles. You have xylem and phloem. Xylem is for anybody?
Water transport, sir.
Yes, transport of water, and phloem is for transport of food. What is the tissue that is present between xylem and phloem? Anybody? Okay, it's called as cambium. Okay, that is another meristematic like tissue which keeps producing more and more xylem and phloem every annual cycles. Annual rings form, right? So the growth of now the growth of plant is from the extremities, as someone said. I couldn't see who is that, but good. And compared with the growth of you, human being, you are small, our hand was very little, and now we are about 20 years old and we have grown this big. And how much does our hand look like? Our initial hand was here. Our elbow is now here. Previously, it would have been here. The growth is not from extremities. We consume food, and growth is all over the body, right? So I just wanted to put this in perspective so that you understand how growth happens. That is where that is also a part of regeneration, right? Plants, if you break away branch, it doesn't bother it much. It doesn't have something like the essential organs like we have a heart, kidneys. We have many things that we require. Even if you take away all the leaves, maybe there is a chance that they can still regrow. Plants have that regenerative capacity a lot. And if you take in plant tissue culture, if you are going to study anywhere, if you studied biotechnology, maybe in plus two, you would have read about it. You can take any part of a plant, be it leaf or root, stem, that's living cells. Majority of them you can take and put it into nutrient media, nutrient agar media consisting of plant hormones, auxins, cytokinins, gibberellins, and so on. That part is called as an explant. A part that is sterilized, you sterilize it of every other bacterial and fungal infections, and then you grow. If you put appropriate concentrations of these plant hormones, the explant, from the explant, some of the cells will grow and you form something called as callus. It looks like this, greenish, a very light greenish yellow mass of undifferentiated cells. Now you can take that callus and make it into smaller pieces and subculture them. Then you're having more of callus. And you can take those media again and put it in something called as rooting medium. That means the callus will generate roots. That is by changing the proportions or the composition of the plant hormones. The ratio matters. I can't remember all the details at this point. So roots will develop. After that, you change this to shooting medium. It already has generated roots, and now it will develop its own shoot as well. You took a small piece of a plant, and you are able to generate a whole organism, right? That is the regenerative capacity of a plant, for example. So plant cells, majority of them have incredible regenerative potential. They can give rise to, of course, I will want you to remember this. All this part is human intervention. Typically, we wouldn't want human intervention. Say, for example, you know the grafting and other techniques. You cut a branch of a plant and put it into soil, or if it falls by itself, it will grow roots. Some of them, not all of them. And they can become an independent uh, organism, asexual forms of reproduction. Some of them, in some cases, the branches, if they touch the soil, they grow roots there. And by any means, if it gets cut here, this is a separate individual, and this one, if whatever is here, will be a separate individual. That is the potency or plasticity, the ability to give rise to other plant parts or whole body itself. So in today's class, we are mostly going to talk about this ability of cells to give rise to different things. Can it give rise to other cell types? Can they give rise to the whole organism or not? And that ability is or the degree of differentiation, the ability to how many differentiated types of cells a cell can give rise to, is referred to as plasticity. Or you can also think of it as potency. There are 4 important words, or 5 totally, but you try to remember these. This will recur. Totipotency, pluripotency, multipotency, and unipotency. I hope you can understand from this. Uni means one. Potency means can give rise to only one type of differentiated cell. That is unipotent. If it is many, this is multi is many. There is also oligopotency. We'll just mix it up. Multipotency means it can give rise to many differentiated cells. Pluripotency means all types of differentiated cells. And toti is the whole organism. There is a difference between totipotency and pluripotency. Both are forming all types of cells that are present in a body, but totipotency is the ability to give rise to whole organism, whereas pluripotent stem cells cannot do that. Okay, we'll get to these. We will repeat this several times, so your understanding or memory would be better. So there is a small history of it. I think when I share the slides or in the textbook itself, it is already there. You might want to have a look at it. but we started from human cord blood. Stem cells were discovered in human cord blood cells from then in 1981, developed from mice, then hamster, and so on. Right now, there are so many cell lines, stem cell lines, including human embryonic stem cell lines that are present everywhere. And some of the hospitals or clinics, they have something like stem cell banks. That's preferably, mostly some of the those that are giving a birth to a child, especially through C-section, because through natural birth, when the baby has to come through the vaginal canal, there is a chance of or increased chance of contamination, the microbiota that is present around in and around the vagina. So if it is C-section, it is typically you cut the uterus and take out the baby from the other source, this side, so there is not as much contamination. So the placenta is collected and it is sent to the stem cell bank, where from there they take out the cord blood stem cells, and they save it for you upon you paying some money. I'm not sure how much it is, but they save these stem cells in the freezer for a long time. I think it may cost something like 60,000. And if the baby ever requires any stem cells, the promise is that they will supply the stem cells to the baby. Okay, so in case of injury in the future, when the regenerative medicine really takes up and is approved for at least some of the diseases, then the child may have access to his own cells that are related to his own system, and they are much more well accepted. So there are two essential properties of stem cells, all stem cells, or you can talk it off as what is meant by stemness, right? There are two properties. One is to, as we discussed in the one of the previous slides, it should give rise to one cell is there, and it should give rise to a differentiated cell, like this. That is, first, one of the properties differentiated. And there is another property, that is, it should give rise to its own type. Say this is morphology or functionally, it looks cell type A. It should be able to give rise to cell type A, and should be able to give rise to some other differentiated cell type, that is cell type B. The previous is called as, this property is called as self-renewal. Okay, and the other property is differentiate. Here I have two examples. I'll say X and Y. If you compare these two, uh, in Y, progenitor cell is giving rise to a specialized cell. Myeloid progenitor cell is giving rise to a neutrophil, and also is giving rise to red blood cell. But is this progenitor cell a stem cell? If we look at it, it is giving rise to, say this is a myeloid progenitor cell, and it is giving rise to two specialized cells, so differentiation is there. That property of stem cell is achieved or fulfilled. But is there self-renewal? There is no self-renewal. Therefore, progenitor cells are not stem cells. Here you take the hematopoietic stem cell, from which the progenitor cell also has come about. There you see that this hematopoietic stem cell is giving rise to a neuron, or it is just not the example directly neuron. Say it would have been the red blood cell or neutrophil. Just for example, you take it. Just that it is a differentiated cell. And it is also able to give rise to a stem cell, and that is self-renewal. So therefore, both the requirements are fulfilled. that is the differentiation and the self-renewal. So that is a stem cell. One thing you would have noticed is stem cells could undergo asymmetric cell division. What is asymmetric cell division? That is one cell type, A, for example, upon division is giving rise to B and B. This is symmetric. It is differentiation and symmetric. If it were giving rise to A, the product cells, the daughter cells, if they are different, this is different from the B. A is different from B, so that is asymmetric. Okay. There are many other cells which can show symmetric cell division as well as asymmetric division. At least you can say that stem cells are the ones which can give rise to one of the product will be asymmetric. One of the one of the product will be the self-renewal. It will be of the same A and A, and the other one could be A, or it could be B or C or any of the other cell types. Okay, try to understand, and we will repeat this over time, so your concept should be good. It's clear and simple actually. Self-renewal and differentiation. I want you to know what is meant by both these things. Formation of the same cell type is self-renewal. Formation of a differentiated cell is differentiation. Only stem cell can do both these things. And now into the essence of this lesson. It is about, this chapter is about embryonic stem cells. And then we will try to learn, as I already described, what is self-renewal and what is differentiated. And every cell is supposed to have a function, and there is a structure function relationship to the cells as well, right? In all of biology, structure function relationship is there. You take proteins, you can take nucleic acids, you can take any of them. There is their functions. If to perform a function, certain structure is required, and that is how all biology works. Look at your hand right now. Why is it designed this way and not like a feet? Because you have certain functions to do with these. You need the agility of your fingers. And to work with minute, smaller things, you should have had freely, independently movable fingers, right? So the same way, every cell in the body is supposed to have a structure function relationship. You must have studied about neurons. They have a certain type of function, and they are long with a lot of axons and so on, because they have to conduct the electrical impulses. There are red blood cells, and they have a certain shape, and they should be something like this, right? Because they have to increase the absorption of oxygen and so reduce the wherever the exchange of gases should happen more. In that sense, the embryonic stem cells are said to be unspecialized. They do not have any function as such. Their only function, if there is any, is to differentiate and give rise to other functional cell. That is their only function. Okay. So from zygote, upon fertilization of oocyte, we get zygote, morula, and then a blastula. In the blastula, we have these, that's called as ICM, or inner cell mass. That inner cell mass, if you take them out, those are nothing but embryonic stem cells. Okay. Typically, you can think of it like this. ICM is equivalent to embryonic stem cells. ICM, you call them ICM as long as it is in the blastocyst. Once you move from the blastocyst into Petri plate or other kinds of growing conditions, media with other growing conditions, then you call them embryonic stem cells. The important thing is that they have some unique properties that we will discuss now. So the first thing, self-renewal and differentiation, they're common for all stem cells. The one difference about embryonic stem cells is they can give rise to ectoderm, mesoderm, and endoderm. If you remember, sorry to go before, I think I should have had that here. If you remember this, in this slide, I have described about once a cell becomes ectodermal type, it can only give rise to certain types of cells, like outer surface skin, central nervous system, and so on. The same way is with mesoderm. Once a cell has differentiated into a mesoderm, its developmental lineage is defined. They are committed to becoming certain cell types. Okay? And if there are any cells, if there is, what is unique about embryonic stem cells is that they can give rise to a cell type that may belong to the ectodermal lineage, mesodermal lineage, or even endodermal lineage. Okay? So that is one of the important properties, how embryonic stem cells are different from the rest of the stem cells. No other cell can give rise to all the three types of these cell lineages belonging to the primordial germ layers, all the primordial germ layers. There are also other morphological characteristics. One of them is they have high nucleoplasm to cytoplasm ratio. That means to say that the nucleus size and the cytoplasm here, nucleoplasm or the cytoplasm is less in comparison to or relative to other cell types. Say for example, majority of the other cells, they have a function, right? You have hepatocytes or nervous tissue. They have hepatocytes should what do you call? You have to reduce, detoxify any of the toxins that may have come. You need to perform a lot of biochemical functions of those. Then you need to have more of cytoplasm. And nervous system also the same case. It has to have along these axons and so on. As I said before, embryonic stem cells do not have any function other than to give rise to. They are just there. They have to absorb nutrition and divide further. So there is one more thing. There are some more things like the cell surface antigens. You just want to remember them. I don't need you to memorize all of those. But as we will discuss in the future classes, there are some factors, OKSM. O for OCT4, K for KLF, S for SOX2, M for CMYC. We will get to those later. These are transcriptional regulators which maintain the cell to be in the embryonic stem cell stage. If these transcriptional factors are not activated, then the embryonic stem cell usually divides and becomes something else, or differentiates and becomes some other lineage type. So that is another property, that you have high expression of these OKSM or pluripotent factors, you call them. Embryonic stem cells are pluripotent. Only zygote is totipotent in animal systems. The other one is called as telomerase. We have read about telomerase a lot of in the previous semester in the course. I hope you remember some of it. In every chromosome, you have centromeres and you have telomeres. Telomeres are at the ends of DNA. I would suggest you also to look at end replication problem. After each round of cell division, each round of replication, the DNA becomes shorter and shorter because the ends cannot be replicated effectively. And that is the function of telomere or telomerase to synthesize telomeres. Telomeres will not allow the loss of the genes that are present on the chromosome by protecting these ends from exonuclease activity. They also solve the end replication problem. There is another thing is they prevent chromosome attachment, fusion. because they are telomeres, the fusion is not is or recombination is prevented. So why should there is one more property of embryonic stem cells is the number of times the embryonic stem cells can divide. Theoretically, these are thought to be as kind of immortal, meaning that as long as you provide the media, they can continue to divide for a long, long time. Okay? If that is should be true, then the end replication problem will be an issue, right? What is the solution for end replication problem? That is to have telomeres. And if you want telomeres to be long after each time, then you should have had telomerase, high expression of telomerase. As you will study in some of the courses in the future or current ones, even cancer cells, they can divide innumerable times continuously. That means by default or in theory, they should also have high expression of telomerase, and that is true. Many cancers, you will find that telomerase activity is very high. And that is why some of the companies will try to treat, add inhibitors and target the telomerase activity so that the cells will eventually die out after certain rounds of replication. Okay. And there is another way of finding a teratoma. What is a teratoma? Teratoma is a tumor. And how is this tumor different from other tumors? Is that this tumor has eyes, skin, tooth, teeth or tooth, hair. It has different types of tissues, all that are arising from all the three primordial germ layers. So let me repeat. Teratoma is a tumor which consists of tissues belonging to all types of primordial germ layers. Okay, so this is a way to determine if a given set of cells are embryonic stem cells or not. We will discuss that again when we are talking about about the techniques involved in characterization of embryonic stem cells. We will discuss there at that time. Okay. So otherwise, what we are trying to, or why we are studying embryonic stem cells, or why they are important, is that they can give rise to their potency or plasticity is the largest possible. They can give rise to ectodermal cell lineages or mesodermal or endodermal and even germline cell lineages. And they can be uh, used to treat diseases. Okay, they are not at any uh, approved medications or therapies based on stem cells. They are all under clinical trials. Okay, but this will be the future of medicine if everything goes well. There are also, in our body, we have adult stem cells. Adult stem cells, you can think on hair or intestinal epithelium or any of the majority of the tissues have stem cells. It's like this. Assume this is an organ, right, in our fully grown adult human. And there should be some 1,000 cells, assume. And then these 1,000 cells should perform a function. And every function will have wearing and tearing. That means the cells get old and cell death. But if this is the case, because this organ is has 1,000 cells and is performing a function because of which eventually it will die, some of the cells, the cell number will go down. So every organ or majority of the organs are embedded with what you refer to them as adult stem cells. Cell production, I will say. Cell production will keep on adding more and new cells, and upon functions, some of the cells will die out. and the number of cells is maintained constant like this. So it is a balance between rate of cell production and rate of cell death. Okay, so if how is this rate of cell production maintained, or what cells are responsible? Those are called as adult stem cells, or you can say it as ASCs, adult stem cells. These are different from embryonic stem cells, because if you take in the hair follicle, there is a bulge, you call it hair follicle bulge. That is where the stem cells are present, adult stem cells, epidermal adult stem cells. They provide the cells required for the growth of the hair and also for the epidermis. You know that epidermis is, the cells will keep on dying and you need to keep on producing new cells. And those are coming from these epidermal stem cells, right? So there is a continuous replenishment, as you can see in this case. Now I want you to also have a little bit of idea. Say I will redraw. This is 1,000 cells, and this is rate of production, and this is the rate of death. If there is increased production beyond requirement, what would you expect? The cells might become more than required. You can also see a similar scenario where the cell death has decreased while the rate of production is normal. Then too, there is a possibility of increased cell number than required. That you can think of it as scenarios of cancer, right? Cancer is something like you have more number of cells than required, right? And of course, it is because of mutations and so on. But maintaining this cell number homeostasis is the essence of every tissues in an adult human. And for that, every organism, every organ system, they have adult stem cells and they keep maintaining the cell number in proper. The same is the case with the gastrointestinal epithelium, where the crypt cells will keep on producing the different types of cells that line the intestinal epithelium. And because intestinal epithelium is constantly eroded or damaged or worn and become old or so, so there should be a way to replenish them, and crypt cells or these stem cells keep replenishing them. I think I'll take the next one later, but I want to leave you uh, with a kind of imagination, if you can think of it that way. This is zygote, and that gives rise to eventually ICM and primordial germ layers, sorry. And then we are grown, an infant, and eventually a complete adult, right? You can think of something giving rise to more number of primordial germ layers, and this one giving rise to different tissues. Here is another tissue, and here is another tissue. On a time scale, if you keep following what is happening to a zygote, this is what is happening, eventually giving rise to a body. You can also see it this way. You have been fond of crackers in Diwali. I don't know what you call them. You shoot one thing, it goes up. That bursts into several other tiny bombs, which will again burst and gives rise to some beautiful flowers in the sky, right, during Diwali. This part, you can think of it as the stem cells. That's what they do in our body every day. If this is the zygote, you can think of, which has part of different tissues. These you can think of as adult stem cells. They keep pumping more and more into the functional, uh, differentiating into the functional cells, right? Okay, I will discuss the rest in the next class. If you have any questions, I will try to answer now. Anybody has any questions? Let me stop the recording.